All right, everybody, welcome back to another episode of Biology with Mr. Emond. Today we're going to be continuing on with our unit on cell division. This is the last lesson in the unit. We're going to be looking at what causes cancerous cells. And let's get started. So here we're starting off with the first understanding that mutagens, oncogenes, and metastasis are involved in developing primary and secondary tumors. We're going to be defining these terms here shortly. Let's start off with coming up with a definition of tumors. So tumors are abnormal cell growths, and they result from uncontrolled cell division. So when mitosis is happening uncontrollably, and the cell begins dividing too quickly, they lead to abnormal cell growths that we call tumors. And these can happen basically in any tissue or organ. Then we need to talk about some of these tumors cause disease. So diseases that are caused by the growth of tumors, that's what we call a cancer. Okay? And there's many things that can cause the growth of tumors which lead to cancers. The first one we're going to talk about right now are mutagens. And mutagens, you're going to probably hear this word a lot, especially if you take workplace safety courses. Mutagens are things that are able to actually change the genetic material of the organism. So they're able to actually mutate the DNA or RNA of the organism, and this is going to cause some pretty drastic changes in the way that those cells function. So there's three types of mutagens. There's physical, chemical, and biological, and this depends on where they come from, so their origin. When we're talking about physical mutagens, we're usually referring to X-rays or UV rays, ultraviolet rays, or radiation, so radioactive decay. Chemical sources, chemical origins, this usually involves reactive oxygen species, so ROSs, and metals. So there's some chemicals, specifically in these two groups, metals and ROSs, that are known to cause cancer. And whenever you see a product with this symbol on your screen, it's a mutagenic thing. So if you're holding something that has this on it, I'd be very, very careful with that. And the last source is biological. So there are some viruses and some bacteria that can, and some transposons that can actually change your DNA. Okay, and anything that changes your DNA is called a mutagen. Not all mutagens cause cancer. Some mutagens, the ones that lead to the formation of cancer, we call them carcinogens. So if you've seen that word before, a carcinogen, that means that it is a mutagenic agent. It's one of these physical, chemical, or biological mutagens that will change your DNA and will result in the development of cancer, a carcinogen. Now we're going to talk about oncogenes. So an oncogene is a gene that has the potential to cause cancer. So genes are things that you inherit, right? This is not the result of being exposed to something. This is something you're born with. So most cancers that fall in the category of an oncogenetic cancer are caused by mutations in two basic groups of genes. These groups are your proto-oncogenes and your tumor suppressor genes, and they work very differently. Your proto-oncogenes, what these genes, these portions of your DNA do, is they code for the creation of proteins whose job is to stimulate the cell cycle, promote cell growth, and cause cell division to happen. Now those, of course, they're very necessary processes in cell division, right? We talked about them in the last lesson. The genes that cause cell division to occur, that stimulates it or allows for the events in the cell cycle to happen, those are normal. What we're talking about right now is when you have a mutation in those genes that causes your proto-oncogenes to basically go ballistic. When a proto-oncogene is mutated, what ends up happening, or like in other words, you have an increased expression of it. So these are genes that cause cell division to happen. When it's mutated and it leads to cell division just happening way too quickly, it becomes cancer causing. So proto-oncogenes, they're not bad things, but when a mutation occurs in your proto-oncogenes that leads to it being expressed at a level that it shouldn't be, it's like hitting the gas on a vehicle and accelerating out of control. So looking over here, if you have a mutation in your proto-oncogene and that leads to you like burning nitrous instead, you're going to be spiraling out of control speed-wise, and cell division is just going to be totally uncontrolled, happening too quickly. The cells might be deformed as a result of that, and this could lead to cancer. So a proto-oncogene, you can almost think of, you're putting the pedal down too far and you're spiraling out of control in that way. Let's look at tumor suppressing genes. So if you have a tumor suppressor gene, what these do is they generate proteins. Remember, genes generally code for specific proteins. These proteins, their job is to repress the progression of the cell cycle. So they're like the brakes. When you have tumor suppressor genes, and it's a good thing you do, it puts a stop to the cell cycle, right? And it slows it down. It also promotes apoptosis, and apoptosis is something that we talked about in previous lessons, especially in Health Science 20. This is programmed cell death. So when a cell, it's time for it to die and we're going to replicate a new one to replace it, that is apoptosis. Tumor suppressor genes are sometimes called anti-oncogenes. The oncogenes are like hitting the gas, and the tumor suppressor genes are like hitting the brakes. So they're almost seen as anti-oncogenes. They slow down cell division because their normal function prevents cancer. So you might be thinking, how can you have tumor suppressor genes that cause cancer? Well, we're talking about not normal, healthy tumor suppressor genes. We're talking about mutated forms of those genes. And if you have a mutation in your tumor suppressor genes, what's going to happen is your brakes don't work. And so your tumor suppressor genes you're supposed to be able to use those genes and create those proteins to slow down cell division or pause it for a period of time. And if you have a mutation in those genes, you have no brakes. You can't stop the car. Okay? So here you have tumor suppressor genes that are inactivated. They've mutated. It's like not having brakes. So even if the speed is normal, with no brakes, you just keep building speed and accelerating out of control because there's nothing stopping you. Let's look at metastasis now. So metastasis is a word that refers to the spread of cancer from one location to a new location. Now, tumor cells in general, when you have a clump of cells that we'll refer to that have uncontrolled cell division, these tumor cells, they can either stay where they are, right? Let's say you have a tumor that develops in your left breast. Well, if it only stays in your left breast, we would call that benign. Or it could spread and invade neighboring tissues. So if that clump of tumor cells somehow spreads in your body and you have a breast cancer popping up in different locations around your body, And it doesn't necessarily have to be in your breasts. You can get breast cancer of the kidney. And we'll talk about that in a second. If the breast cancer, the location of the original tumors, spreads or invades any neighboring tissues, that's when we start calling it a malignant tumor. So benign tumors just mean that the tumors are still in their original location, and malignant means they've now spread. We can also talk about them in terms of primary tumors and secondary tumors, right? The primary tumor is the location is the tumor that originated in your original location, and secondary tumors are new tumors in new locations. The interesting thing, I don't know if interesting is the right word, but the interesting thing about secondary tumors is they're made up of the same type of cell as the primary tumor, and they can occur in different parts of the body. So for example, you could have breast cancer spread to the liver, and therefore we would say that that patient has secondary breast cancer, meaning it's a secondary tumor, right? The type of tumor is breast cancer, and it's in a new location. It's secondary breast cancer, and this is breast cancer of the liver. And how do you treat breast cancer of the liver? Well, you treat it the same way as breast cancer of the breast. You treat it with breast cancer drugs, okay, or breast cancer treatment. Let's look at this diagram here to kind of explain how this happens. Let's say that you have a group of healthy, normal cells, and something happens during cell division, and we have a mutation in our DNA, a mutation occurs in our DNA, and all of a sudden we have a cancerous cell. This cancerous cell is going to start replicating. So you can see our one unhealthy cell starts to replicate. The rate at which this unhealthy cancerous cell replicates is going to be much faster than the normal, healthy, non-mutated cells. It's going to start to overwhelm the area, and a primary tumor is going to develop. This primary tumor is going to start developing vascularization. So this means that blood vessels are going to be forming around these cancerous cells and turning it into a structured region in your body. And blood's going to be flowing through these cancerous cells. Now, something that can happen is you can have a single cell, a cancerous cell, detach and intravasate, or move through the blood vessels and travel through your bloodstream, exit your blood vessels, extravasate, and invade a new area of your body that has only healthy cells. When this happens, this is how you form a secondary tumor. Okay, so that tumor, those cancerous cells have then spread to a new location and have formed a secondary tumor, which again can vascularize, and one of the cells can then move on and cause a tertiary tumor. Let's compare normal cells to healthy cells. So first we'll go through here. Cancer cells can commonly avoid immune detection because they're not actually foreign bodies, right? They sometimes usually look like your own cells. They're just functioning abnormally. And so often your immune system doesn't detect them and flag them as being invaders that need to be destroyed. And this is what makes them difficult to treat. And so some of the strategies to remove tumors especially are removing them surgically, and sometimes we'll go through chemotherapy or radiation therapy. Let's look at the differences between normal and cancerous cells now. If we look at this normal cell here, it's small, it's uniformly shaped, and its nuclei is also uniformly shaped. And if we look at the difference in size between the nucleus and the whole cell, there appears to be a pretty large cytoplasm here. Looking at the cancerous cell, it's going to be larger, and it's not going to be uniformly shaped. It's going to be variably shaped. And the cytoplasm is going to be much smaller because the nucleus is larger. look at the actual cells here. So in normal cells, there's some conformity between them. They have similar shapes and similar sizes. In cancerous cells, like I said, they're going to be different sizes and different shapes. Their normal cells are also going to be arranged in discrete tissues. So you'll have some cell types that only belong in certain tissues. In cancerous cells, the organization of them, they're not very organized, right? They're disorganized in terms of their arrangement. They're not going to be uniformly like kind of forming a line like these are. They're going to be kind of all over the place. Normal cells have different cell structures within them and on the surfaces, right? We can have proteins on the surfaces, and they're going to present normal cell surface markers. So the proteins on the surfaces, the peripheral proteins on the surfaces are going to be normal, and in cancerous cells, you're probably not going to be able to function. So the organelles within your cell you might not have those cell structures because your DNA is now mutated and not functioning properly. You also, the markers on the outside are going to be unhealthy. They're not going to be what's supposed to be there. Okay? So some cell markers are going to be expressed at higher levels. Normal cells don't divide very fast. They have lower levels of dividing cells. Cancerous cells divide very quickly. And so you'll have a lot of cells dividing very frequently. And cell tissues in normal cells are clearly demarcated, which basically means that there's a clear boundary that the cells exist in. And cancerous cells have poorly defined tumor boundaries. The last thing we're going to talk about is the correlation between smoking and cancer. So if we look at graphs where you have the amount of cigarettes smoked per day on the x-axis and the amount of cases of cancer occurring in the population, you'll notice an exponential curve here, where as the amount of cigarettes increases per day, the amount of cancer in your population increases. And we can look at this second figure here. This is a little bit different. On the x-axis, we have the amount of cigarettes that are being manufactured per country per adult. So this would be manufacturing 500 cigarettes per adult, 1,000 and 3,000 cigarettes per adult. And on the y-axis, you have the incidence of lung cancer between the ages of 35 to 44. Now, this is an interesting chart too. When you graph the countries, there seems to be a clear correlation. If I was going to draw a line of best fit, that's probably a bad one. If I was going to draw a line of best fit, it might look something like this, right? It seems clear that as a country produces more cigarettes, lung cancer develops. And there seems to be a correlation between the amount of cigarettes that are produced per capita and the incidence of lung cancer. There's numerous studies that highlight the correlation of lung cancer with smoking, with roughly 90% of lung cancers being attributed to tobacco use. And some of the new research is going into e-cigarettes or vape pens and is trying to determine whether or not these vape pens also are carcinogenic. All right, that's all we have for today. And that is the end of the unit. Thank you very much to our sources of information, BioNinja and the Oxford IB Diploma Program textbook. For homework, if you go into our Organization of Life folder and go to cell division, you will find the video will be posted here, and there is a cell cycle and cancer virtual lab. This is what I would like you to try to complete today. You can click this link and make a copy for yourself and just follow the instructions and answer the questions embedded in this. So thanks for watching, everybody. You can like, follow, and subscribe to my social medias to get notifications as soon as new content is uploaded. In the top corner here, you will find the playlist that this video is in. Watch the unit from beginning to end, and over here you will find the next video in this unit. Keep learning, everybody. Take care.